So I'm delighted to be chairing this particular session. My name's Kath Mummery, and I am an Alzheimer's trialist at Queen's Square in London. And I think right now we're really entering a new era in treatments in neurodegeneration. So it's really exciting to have a session dedicated in this horrific disease to potential new therapies. We've got a fantastic group of speakers lined up for you and an awful lot to pack in. So I am going to introduce our first speaker without any further preamble. Speakers, as a visual aid, I will put my camera back on 2 minutes before time. I'll give you an alert 1 minute before time. And the first two speakers have 10 minutes. The third has 15. Do please put questions in the Q&A as we go along. And please add who they're directed to so that we can get the right person to deal with them at the end. Thank you very much. So the first person up is Professor James Rowe, good friend of mine who leads the Cambridge Centre for Frontotemporal Dementia and Related Disorders, including PSP, is a principal investigator in the Cambridge Centre for Parkinson Plus, and is a trustee of the PSP Association. His research promotes development and validation of disease-specific biomarkers and therapeutic strategies in PSP and CBD, and he'll be talking to us about NORAPS, a new potential treatment in PSP syndromes. James, over to you.
Thank you. Can you hear me?
Perfectly.
Thank you. Fantastic. Thank you. Well, thank you, Professor Mummery, for the very kind. And I'd also like to thank Annalisa for organising such a fantastic symposium today. So thank you.
James, have we lost you? Yes, I've got a few nods. Maybe if you turn your camera off, we might get a better reception.
Right, so let me just try once again. I do apologise.
No problem. We can hear you now.
I'm hoping, can you see my screen?
Yes.
Fantastic. Thank you, Professor Mummery, for the kind introduction, and to Annalisa for organising such an event as today. It's really very exciting. I've enjoyed the morning, and even more to come this afternoon. I'm going to talk about the NORAPS study, way of treating symptoms in progressive supranuclear palsy with noradrenaline. And the challenge that we faced was that PSP so often causes apathy. It changes one's motivation, the effort that one can make, the initiation of conversation or activities, or in everyday terms, one's get up and go, gets up and goes. And it's really a psychological change. It's not to do with the immobility in practical terms, and it's not to do with depression or mood. It's a more fundamental lack of oomph and motivation. But PSP can also cause impulsiveness and impulsivity. This is perhaps less often talked about. One might see it if people are reckless. They might get up and stand too quickly, not waiting for someone to give them a hand. It can be seen sometimes, you may recognize, in people eating too quickly, leading to more choking. And sometimes people may speak out of turn and be rather blunt or direct. Now, from a layperson coming to these problems of apathy and impulsivity, you might imagine they're the opposite. One's about being too slow and doing too little, and the other's about being too quick and doing too much, a sort of Jekyll and Hyde of the brain. And this turns out not to be true. Actually, the people who are more apathetic are more likely to do things that are impulsive. Very curious, you can get this combination, but we see this in PSP and corticobasal degeneration and a number of other illnesses. So this is a result from a study of ours looking at a couple of hundred people. And some people are high on apathy and impulsivity, and someone else has neither apathy nor impulsivity. These two things go together. So what's happening in the brain? What drives these two problems? And why does it matter so much? Well, one of the results of the Pippin study, the earlier phase of the Pippin study, was we noticed that people who have more or less apathy have a really different outlook. So the top third of people with PSP, in terms of their apathy versus the lower third, it has an extraordinary difference of 18 months in the time to either needing nursing home care or succumbing to the illness. So 18 months difference in what is already a very rapid illness, I think it is a huge impact of these two symptoms. And we've seen this both in PSP and those related illness and with apathy. And this what we call independence, the ability to live at home with your family are both affected. So it's a huge problem. and one that matters for individuals. But biologically, what's causing it? Well, from a wealth of background science, we know that apathy and impulsivity are both controlled by a chemical called noradrenaline. It's a bit like the brain's adrenaline, and it's made from one tiny area at the back of the brain called the locus ceruleus. It's a very, very small area down here at the base of the brain. It's a bit the size of two short lengths of spaghetti. So I put up on the right-hand picture just a little indication of how small this is. And that sends branches and connections to supply the whole of the brain with this essential noradrenaline. Now, if we look at people who have kindly donated their brain to research through the brain bank, and we zoom in under the microscope, on the top right here, we see a little area. It's about the size of a head of the pin under a microscope. And can you all see these black dots, those little black dots? Each of those black dots is a cell that's making noradrenaline to be sent off to hither and yon across the brain. And there's lots of them. If we look at somebody who's had PSP, you can see a few dots, but there's hardly any left, just indicating how devastating that PSP can be on this critical, albeit small, part of the brain. So this creates an opportunity. Perhaps we could restore the level of noradrenaline, improving the work that's done and the performance of those surviving brain cells in order to improve those challenging and difficult problems of apathy and impulsivity. And we know from a drug such as atomoxetine that we can increase the levels of noradrenaline safely and in the brain. This is a drug that's widely used now in ADHD in children. It's not normally used in older adults, but we know that it chemically does what we would want to do to boost the levels and recover noradrenaline. And over the last 10 years, we began working with Parkinson's disease, and over a series of studies, we could show that we could improve in impulsivity, what we call response inhibition, in people with Parkinson's disease. And we could work out which individuals were more likely to get a good response versus some people who don't respond. This then came forward into what we call neuropsychiatric symptoms, including apathy. But a landmark study came from the use of a very similar drug called methylphenidate for treating apathy in people with Alzheimer's disease, another illness that affects the noradrenaline capacity in the brain. And recently we put together all of the studies of noradrenaline type treatments and showed that whether it was atomoxetine or other drugs like methylphenidate, together they would improve apathy in these brain illnesses. So that really gave the impetus to this study called NORAPS. This is a phase 2 study, which means a medium-sized study. We're looking for 80 people. And it's looking for both the safety and tolerability of the drug in people with the illness and early signs that it might be helpful on symptoms. So I'll just talk a little bit about who and what the study is, so you have a flavour of as it unfolds. Well, the people we're aiming for is really anyone with PSP. We've designed the study to be as inclusive as possible. Whatever personal variant of PSP, and whether you're mild or moderate or more severe, we'd like this to be something that could be possible for you. Not everybody can take these medications with other illnesses and other tablets they might be on, but we reckon about up to 3/4 of people, in principle, might be eligible. It's currently set up at the initiating sites in Cambridge and Southampton. We hope to bring Newcastle, Glasgow and Salford online soon. And there's some other sites in the UK, perhaps nearer to you where you're listening from later in the year. I won't name until I know that we're a little bit further down in the setup. We would like this to be within reach of as many people as possible. Now, in terms of the tablets, we're actually not using pills, we're using a syrup. We know swallowing can be an issue, so we're going to use a syrup of the atomoxetine that's very easy to swallow. And we're comparing the response to the real drug atomoxetine to a placebo. And some people worry about a placebo because it's a dummy pill without the real stuff in. But we're doing what's called a crossover design. So you might get atomoxetine first and then placebo, or placebo first and then atomoxetine. The idea being that everybody gets a crack at the treatment. We get to see in everybody whether it seems to help over a few months of treatment. And the potential we're looking for is to know that it's both safe and well tolerated, but also giving some clues as to its efficacy, whether it's improving signs or symptoms of apathy and impulsivity. Now, we don't expect this to work for everybody. Certainly the work from Parkinson's tells us that some people may respond very well and others not. And we want to, in the long run, perhaps move to personalized medicine, getting the drug to those who would benefit and not treating those who we think are unlikely to benefit. So how might we do that? Well, we've learned over the last few years that atomoxetine works best in other diseases in people where there is most severe damage to the noradrenaline capacity. So where people have more damage to this locus coeruleus, this tiny spot at the back of the brain, and it's, post-mortem where people have donated their brains to research, if we look directly, we can see this little blue spot about the size of the head of a pin. That's the locus coeruleus, but that's at post-mortem. How can we see this in people early in the illness when they're very much alive and coming to clinic? Well, I've shown you those cells that we can see under the microscope, and they're black under the microscope because they contain lots of iron. If we zoom in onto them, there's lots of iron, tiny microgranules of iron in those cells. And that has enabled us to develop a special variation of MRI scanning where we can actually picture this noradrenaline producing part of the brain, seen here as a little red dot corresponding exactly to what we see under, in life. So with that, we can then measure how much noradrenaline capacity you or an individual person has. And we're hoping that this noradrenergic will not only treat apathy and impulsivity, but we'll be able to understand that individual difference in efficacy based on your noradrenaline capacity. So the next studies would be able to target more specifically. Now, if I just think ahead, where am I going as a researcher and as a doctor with this? Well, this is very much a symptomatic treatment study. We're trying to reduce those troublesome symptoms. But we've shown from the long-term studies that apathy and impulsivity have a real impact on the outlook of the illness. They predict how long it might be before you need nursing home support, or indeed the very survival from the illness. So it's just possible that treating these symptoms might also improve the outlook for people with PSP. So with that, I'd like to thank you for listening. I'd like to thank my fantastic team at the Centre for Parkinson Plus here in Cambridge, and for the collaboration in Prospect. Really, this is a national UK partnership, and I think it's a very exciting time as a doctor and a researcher to be part of this PSP community. Thank you.
Thank you, James, for a fantastic talk, and you kept beautifully to time. Really appreciate that as well, because it's such a tight agenda. I've got lots of questions, but we're going to move straight on and keep the questions to the end. So our next talk is from Dr. Boyd Gosh, who is a clinician and a full-time consultant neurologist in Southampton and Salisbury. And he has a particular interest in patients with Parkinson's plus disorders, having previously completed research in these syndromes as part of his PhD in Cambridge. He's going to be talking to us about an early phase trial of a treatment that he will explain more about in a minute in those with PSP. Boyd, I'll hand over to you. Thank you very much.
Thank you. I presume everyone can see my slides?
Yeah. Okay,
so unlike James, this study is a study that's a commercial study by pharmaceutical companies. So my involvement is really to try and deliver that study for the pharmaceutical company rather than the background science. But I thought I would explain a bit of the science behind it and then explain what we're doing. So as Professor Mummery said, I'm a consultant neurologist in the Wessex Atypical Parkinsonian Syndrome Service, and also a trustee for the PSPA and one of the research committee members for the PSPA. So a lot of interest was generated by these headlines in the BBC, where one of my patients, the first patient to trial this drug in the UK, was featured with her permission, clearly. So what were we doing? So you've heard a lot about pathology in PSP, and this is largely, I'm afraid, for progressive supranuclear palsy rather than corticobasal degeneration. But in PSP, we all have, well, all of us have tau protein in our brain, and that helps one of the transport mechanisms in our cells, the microtubule, to carry goods or nutrients from one end of the cell to another. But in PSP, as with some other diseases, sometimes that tau protein becomes disordered and wraps itself up and becomes a filament, and then a big lump of protein which prevents the cell from working properly. And one of the thoughts are that the spread of the disease of PSP across the brain is caused by those abnormal shaped proteins, tau proteins, spreading from one cell to another, And when you get an abnormally shaped protein arriving, tau protein arriving close to normally shaped protein, then the normally shaped protein changes its shape. So it becomes very similar to the abnormal protein and then spreads like a seed, if you like. So that spreading abnormal protein, abnormal tau from one nerve cell to another would spread the disease from one nerve cell to another, and you'd slowly get progression of the disease across the brain. So you can see at the bottom how the disease progresses. So the green areas are abnormal, and you can see at the top how the seeds of abnormal protein tau may spread from one cell to another to facilitate that. So the company said, how could we reduce the progression of PSP? Well, one way that we could do that maybe is if we switched off the production of normal protein tau, or at least reduced it, then there's less protein to change shape, and therefore less ability for abnormally shaped protein to build up in cells and for cells to die. So it may at least slow down the disease. So I'm going to give you a bit of a background on genetics now. So we all have Ed Jabari did some of this as well. We all have in our cells DNA, and let me just see if I can. There we go. So we have DNA, 2 copies of DNA, and when we want to create a protein or an enzyme, we split the DNA into, so we've got only one strand of the DNA. We create something called messenger RNA, which is then translated through this thing called a ribosome into protein. Okay. So we, or the drug company, are using something called an oligonucleotide. And what they do is this oligonucleotide is a very small piece of nuclear material, and it binds to the messenger RNA. And it binds in such a way that actually the body thinks that the RNA, the messenger RNA that would have turned into a protein or would have been translated into a protein is malfunctioning, and it gets rid of it. So it breaks up into small bits and degrades that piece of messenger RNA, and so therefore you don't get the protein because there's no message, there's no messenger RNA to tell the body how to produce the protein. So Novartis set up this phase 1 study, and I'll explain what a phase 1 study is in a second, to test this antisense oligonucleotide. So it's effectively a very small piece of nuclear material which binds onto the messenger RNA, disrupts the message, and therefore stops the body from producing, or limits the body producing protein, the protein tau. So firstly, a few things. A phase 1 study is a safety study. So it doesn't look for how well this works in patients with PSP. It works to see it. The idea of the study is to see, is it safe? So can we give this to people, and do they develop side effects because of the treatment that we're giving them? And so because of that, patients are only given a limited number of drug doses. And because this is a bit of nuclear material, this has to go through into the space around the brain and the spinal cord where the spinal fluid is. And so we have to give people a lumbar puncture. So we put a small needle into their back, inject this medicine into the back, and then we see if they have any side effects. And in fact, most patients on this study will only have 4 or 5 treatments. And unfortunately, the drug company are not planning on doing further studies to look for effectiveness in PSP, because unfortunately, Alzheimer's disease, of which there are many more people with Alzheimer's disease, also has tau protein problems. And so they're switching to Alzheimer's disease to see if there will be a clearer sign. From our point of view, we have recruited 2 people already. We're only due to recruit another 2, and we have already chosen some people from our regional clinic. The limits of the study suggest that we need to make sure that people are very close to our centre in case there is a problem. And that has limited, I know that we've had lots of interest from lots of people around the world even, but that's limited our ability to include people in the study, that together with the small numbers that we're recruiting. So I'm just about finished. So I just want to say thank you to the staff and patients and carers at Wessex APS service, particularly Jade Donnelly and Luke Massey, who are the clinicians that work with me, and without whom I couldn't run the trial. Thank you to PSPA for funding research, which you'll hear about later. And thank you for some of our charities that have funded equipment that we give out to our patients from our equipment library to enable them to try them before they have to buy them, to see if they improve their quality of life and their ability. And thank you for listening.
Thank you so much, Boyd. That was fantastic explanation of what these gene therapies are looking at the moment. And I'm actually about to start that drug in Alzheimer's disease. So useful for me too. We will move straight on, but I hope people can put their questions in the Q&A for later. Even if we can't get to them, we will try and answer them separately. So do please put anything you like in the Q&A for us to answer. But I'm going to move on to our third and final talk for this session. And I'm delighted to welcome Prof. Larry Golb to this event, and that he's going to give us an international overview of what's going on in terms of treatment potentials in PSP. Larry is Emeritus Professor of Neurology at the Rutgers Robert Wood Johnson Medical School in New Brunswick, New Jersey. His research is into the clinical genetics and epidemiology of the Parkinsonian disorders, and he has led understanding of these areas for over 30 years in the Parkinsonian syndrome. So he is a giant amongst men. He devised and validated the PSP rating scale, which is now our standard clinical measure, and is Chief Clinical Officer and Scientific Advisory Board Chair of Cure PSP in New York. So over to you, Larry.
Thank you very much. Can you hear me okay?
Yes, perfectly. Thank you.
I will try to share my screen. All right. How's that?
That's great. Thank you.
Okay. Well, thank you for that kind introduction, excessively kind introduction. As you said, I retired from my professor post 3 years ago at Rutgers Robert Wood Johnson Medical School, but I continue to work as a volunteer with Cure PSP in New York City, an hour away on the train. I've been volunteering with them since the year of their founding, which was 1990. And now that I'm retired from my professor's job, I've actually increased my activity there. It's a very, very well-run organization and very productive. We are, first thing first, my disclosures for the past few years, I've been doing consulting work for a number of drug companies. The two that might be relevant to this talk are Mitacon and UCB. This is just for helping the companies design clinical trials. I don't have any profit motive here. I don't own any stock or have any financial interest in the success of these drugs. I work on some advisory boards, and I have this book, which is not gonna provide much financing for my retirement, I'll tell you that. Now, we're going to talk about treatment. Let's divide treatment into 3 categories for orientation purposes: symptomatic, protective, and curative. So symptomatic means just treating the superficial symptoms, make the person feel more comfortable, perform their daily activities more effectively, without actually affecting the progression of the underlying disease process. Nevertheless, symptomatic treatment can prolong life in important ways, because particularly in a disease like PSP, where a limitation of survival is because of the complications of the disease, such as falls and infections and blood clots. Improving mobility with levodopa can prevent many of those things. It doesn't help the majority of people with PSP, but it's worth a try. Sleeping medications and bladder control medications. A lot of people with PSP will get up during the night to go to the bathroom and they'll fall. So if you can, and break a hip or have all sorts of problems, and a medication that strengthens the bladder, that inhibits the emptying of the bladder, can improve that, and the result is prolonged survival. Now, neuroprotective that's the hot topic. In my talk here, I'll be discussing secondary prevention. Primary prevention prevents the disease from developing in the first place in healthy people. For example, vaccines are primary prevention. Secondary prevention is where the person already has the symptoms of the disease or the diagnosis, and the idea is to slow or stop the worsening of the disease process. But it addresses the underlying disease process in the brain and not just the outward symptoms. And finally, curative is a ways off for PSP, but that would be where you can slow or halt or even reverse the disease process and maybe give it some accompanying approach to repair existing damage, stem cells, growth factors, that kind of thing. So first thing I'm going to do is to kind of show you this cemetery of trials that have failed in the past. These are only the large double-blind trials, not always so large, but these are the double-blind trials that have been going on, starting with, in 2009, riluzole. This is a drug that has been shown to slow the progression of amyotrophic lateral sclerosis, motor neuron disease, as you call it there. It does that only very slightly, and it doesn't work at all for PSP. I'm not going to go through all these in detail. I will point out that some of them address the tau protein directly. You heard an excellent description of what the tau protein is doing wrong from Dr. Ghosh a few minutes ago. This drug, tidegliflozin, inhibits the phosphorylation of tau. That means the tau protein normally is regulated, its activity is regulated by the attachment of this tiny little molecule called a phosphate group. This is a normal thing, but sometimes there's too many phosphates or they're in the wrong spots. And so a big commonality in many of the preventive approaches to PSP is to inhibit the attachment of tau to the tau, inhibit the attachment of the phosphate to the tau molecule. So tidegliflozin does that, valproic acid does that. Then this salsalate, which is just a non-steroidal anti-inflammatory that's approved in Europe, this reduces tau acetylation. So not only phosphate can be attached to tau as a way of normal regulation of the tau, but also acetate can be attached to it by the body. And then there are these two drugs. These are both monoclonal antibodies. They are they famously failed a couple of years ago. They are directed against the N-terminal of tau. What that means is the left side of the molecule as it's conventionally depicted. And I'll get back to that concept in a later slide. And finally, this RT-001, this hasn't been announced publicly yet, but the drug company gave me permission to mention it. This is very interesting. This, if it worked, it would be very interesting. This protects the cell membranes from attack by free radicals, which are toxic little chemicals produced as a byproduct of energy metabolism in the mitochondria. Sorry if those terms zoomed over your head. Free radicals are normally produced by all of us, but we have mechanisms for mopping them up and rendering them harmless, as long as they're produced in normal amounts. In PSP, they're produced excessively, and the mechanisms for mopping them up aren't working so well. So there's too many free radicals. But this drug is a fatty acid that is linoleic acid, which is a common fatty acid in our membranes this is a resistant version of the fatty acid that protects it against the free radicals. It would be nice if it worked. Now, here are the ongoing active trials. Some of them are still recruiting, some are not. This one is just at the University of California, San Francisco. This addresses the phospho-tau. It reduces the attachment of phosphate to tau as performed by this enzyme called Rho kinase. In general, when you see the word kinase, that's a general term for enzymes that attach phosphates to tau, to any protein. So that's fully recruited and will be completed late this year. Still just an early phase study with only 15 patients. This TPN-101 is a repurposed AIDS drug. It's a reverse transcriptase inhibitor. Many of the AIDS drugs on the market that are very effective are reverse transcriptase inhibitors. This one happens to work in a way that would help prevent or slow down PSP. It's being tested in these 5 sites in the US and not anywhere else at present. Also a small study, and that'll be completed in just a few months. This last one here is the antisense oligonucleotide that Dr. Ghosh mentioned. It's still recruiting. Southampton is just one of the sites internationally. Intrathecal infusion, which means it has to be injected directly into the spinal fluid at the base of the spine, as Dr. Ghosh mentioned. And this is because it can't get through the blood-brain barrier, which is basically just a very tight connections between the cells lining the blood vessels in the brain that don't exist in other organs. They're very selective in what they allow into the brain, and they do not allow this large molecule into the brain. And as he said, unfortunately, the drug company is not going to continue development of this drug in PSP after this trial. They're going to stay with Alzheimer's, but maybe they'll get back to PSP eventually. We don't know. Now, more drugs in the same category of active neuroprotection trial. This first one is the one I mentioned before that has failed. I left it here in this slide because it hasn't been announced publicly yet. This one, AZP 2006, is a neurotrophin inducer. That means neurotrophin is a growth factor, a trophic factor. It helps brain cells grow and form new connections with other brain cells. It's given as an oral solution. The trial is going on only in France. Small trial has completed the recruitment. This one here, the bepranemab, is big drug company, UCB. This one it's a monoclonal antibody like the ones that failed, but unlike those which were directed against the N terminal, the beginning of the antibody, this one's directed against the C terminal, which is the right side of the molecule as conventionally illustrated. And the C terminal is where the tau molecule attaches to the microtubules. Now, the microtubules that's basically the brain cell's monorail system. It transports things out to the connections to the other brain cells, transports other things back from the connections to the center of the cell, and also provides an internal skeleton to maintain the three-dimensional structural integrity of the brain cells. So, so if your tau is not working right, then those microtubules might not be working right. So it seems a little more logical to direct the antibody against the end of the tau molecule that actually attaches to the microtubules. And that's what this antibody does. So maybe this one stands a better chance than the previous ones. We will find out. Like any monoclonal antibody, this is given as an intravenous infusion. It gets across the blood-brain barrier, but you can't take it as a pill because it'll be destroyed by the stomach acid. And then the last one in this category of active neuroprotective trials is sodium selenate. This is reduces phospho-tau. This is only in Australia, and that's still recruiting. Now, symptomatic trials, you heard from Professor Rowe about the atomoxetine, so I won't mention anything more about that. There's this actively recruiting trial in the US of these 2 sleeping pills that are on the market, available at pharmacies, but they've never been tested really in PSP. So that's what they're doing. And this is by remote evaluation. The patients don't have to travel anywhere. That's still recruiting. Then these 2 trials of muscle strength testing. One is expiratory, meaning the muscles that help you breathe out. That seems to help, at least preliminary evidence is that it helps the swallowing problem in PSP. So that's being tested in Germany. And physical therapy, exercise just for gait. This is a double-blind trial. It's hard to do double-blind trials for physical therapy because it's hard to disguise whether you're getting the real thing or not, but they're going to try. Then there are these electrical treatments. Transcranial magnetic stimulation is the first 2. This is a device about the size of your hand that attaches to the scalp. It's non-invasive, sends a magnetic field into the brain. This one is a transcranial direct current, so it actually sends an electrical impulse into the brain as opposed to magnetic. That's only in Italy. And then electrical stimulation of the spinal cord in China. There's preliminary evidence that that might work. Finally, in the near future, what's in store? Well, this little drug company based in Switzerland, Acceleron, has a drug that, again, like the phosphate and like the acetate, there's this sugar called N-acetylglucosamine, which attaches to tau to regulate the function, and this drug addresses that. I emailed the head of the company a few days ago. He said the plans are not yet formed for a trial. This one, MP101, is this takes advantage of the fact that there's a problem with the mitochondria, the energy-producing parts of the cell in PSP. It reduces the production of those free radicals. They're planning a phase 1 trial. I'm helping them design the trial. And finally, this tolfenamic acid. This is another repurposed drug. It's an NSAID. It reduces tau production. Other NSAIDs don't do that. And this would only be a Cleveland Clinic in Las Vegas. The trial would be ready to go, but the company doesn't have the money yet. And finally, a big hello from all of us at Cure PSP, all of you at the PSPA and people in the UK and elsewhere who are fighting PSP and their caregivers and families. Good luck to us all. Thank you for listening. I'll be glad to answer your questions.
Thank you, Larry. That was such a terrific and comprehensive overview of the treatments in the different areas that we have at the moment, and a glimpse into the future, which has to be brighter. We have had a lot of questions coming through that I think people have been answering as they've been going through. But if you do have questions, we have 5 minutes right now when we can ask the panelists to take those. So do please feel free to write them in the Q&A. In the meantime, if I might take chair's prerogative, Larry, can I ask you a question?
Yes.
So you've obviously got huge expertise in this area in terms of trials, and going through that, what struck me was 2 things. Firstly, the variety of methods that are being looked at, and secondly, the small number of trials when we look at the numbers that are being studied in Alzheimer's disease, but even more so in something like cancer. Do you think that this is really a numbers game primarily, and that we just need to get more trials? What do you think are the biggest hurdles to getting those trials in PSP?
Well, one thing is that PSP is a much smaller market for profits, of course. And I think there wouldn't be any trials in PSP if not for the fact that it's a tauopathy and that it may be a test bed. Well, it is a test bed for Alzheimer's disease. I think if we can improve our outcome measures in PSP, that would be a big improvement. Right now, the standard is the PSP rating scale, which has its problems, I'll be the first to admit. And but maybe some kind of a PET scan or something else, some chemical in the spinal fluid, such as neurofilament light, might be a better marker, or something electrical. So a marker is a big drawback. Also, since PSP is so rare, it's hard to recruit enough patients in one center. So you have to have a lot of centers. It makes it expensive. It makes it unwieldy to have a trial. So I think those are the reasons.
I think that's very familiar in my experience in my own center. I do think the optimistic point there is that on the back of the more common Alzheimer's disease trials, I think there's an awful lot that can be learned and hopefully can accelerate trials in PSP. At least that would be great. I've got a question here for James. James, are you with us?
Yes.
Brilliant. So this is a question. I'm due to see my neurologist in a couple of weeks' time. Is it worth mentioning atomoxetine to him, as I'm currently very apathetic? I'm currently taking Sinemet and Circadin.
Yes, I'm very nervous about recommending drugs that don't have evidence that they work. And I think for new drugs, ideally they'd be studied in the context of a clinical trial that brings all the safety and regulatory backup with it. Many doctors, we do use drugs in what's called off-license use, and that needs a very careful one-to-one discussion around the risks and benefits. And I don't want to have that on a public forum. I'd be very happy if your doctor wanted to write to me, and we could perhaps talk about some of those risks and benefits to help you and your doctor make the decision between you.
Thank you, James. Great answer. Maybe I can ask you one more question about your trial while we're waiting to see if there are any other questions that come up. What was terrific is it's such a real-world inclusive trial, and it's really nice to see that coming along in the treatment trials that we're doing, because they are also constrained. How do you manage that in reality in the trial? How do you get meaningful results out of it that mean something to the people that are taking part?
So, I mean, the first is to recognize the importance. I've been very frustrated when I look at some of the big international trials. You look at what are the rules for who gets in and who's not allowed in. You might find only 1 in 10 or even 1 in 20 people can make it into the trial. That's very frustrating for the other, 9 out of 10 people. It also raises anxiety in my mind that even if the trial works, we don't know if it works for the real body of people who have the illness. So trying to get trials that are representative and inclusive is really important. And another aspect of that is when members of the prospect team, we've looked at what's the outcome? What happens to people with the illness if you're one of those people that does meet the entry criteria or doesn't? And it turns out the outlook's very different. The sort of people trials are bringing in kind of don't look like your average person with PSP, and we have to do better than that as a field. With the atomoxetine trial, NORAPS, we from the outset said we wanted to be inclusive, not just the classic Richardson syndrome, but a variety of presentations of PSP. And we wanted to be very pragmatic. So we haven't layered on lots of obligatory scans, lots of spinal fluid and weekly assessment. We've tried to keep it very feasible, something that's doable for a typical person with the illness. And sometimes it's hard enough to get out of bed, never mind travel cross country to a specialist center. So we want that pragmatism and that real world flavor. It's perhaps a bit easier for symptomatic trials because of the nature of the drugs we have. But I think really it's just an attitude problem from the trialists. I'd look to Larry to perhaps comment on this and what we as a field should be doing to make drugs come through that are more relevant to the average person with PSP. It's a real challenge.
Larry, you've got the last say, and then we have to wrap up on this because we're running out of time, but please feel free.
Well, I think in talking to a lot of drug companies lately, I think they are more cognizant of the need to broaden their entry criteria beyond just this highly refined, non-demented, not very disabled patients with purely Richardson syndrome. So they do get it, and it's in their interest to recruit more broadly. The problem is they're worried that there might be something unusual about some patients, and they want to get kind of a pure culture in order to prove the drug at the outset, and then they can worry about the quote atypicals.
So a lot of work to do, but it sounds like light is dawning and we're going in the right direction, which is fantastic. And the last plea I'm going to make from one person that's written in is, please, can they see the results of the trials and where should they go to look? So I think we can answer that question. Do you want to do that, James, very briefly, and then we'll stop?
That's an absolute promise. These will be published, whether it works or doesn't, it will be published. In fact, we'll be doing more than that. We'll be making the data available to other researchers to kind of pore over and look at more closely as well. This is just, it's an ethical as well as a sort of legal and governance, I think, obligation. So yes, I think the results must be made available.
Thank you very much.
And they will be, I promise that.
And
thank you, James.
And I'm sure the PSPA, and I know Cure PSP will announce the results of all studies as they come through.
Yeah. So watch this space and you will find out. Thank you very much for some terrific talks, and we will now close this session and move to the next session. Professor Adrian Williams, I'm handing over to you. Thank you so much, everyone.